Hi, I'm Taylor Kuhn, an assistant research neuroscientist from UCLA, and I'll be talking to you today about low-intensity focused ultrasound as a non-invasive cognitive neuroprosthetic, demonstrating early neuroimaging and behavioral findings. As we all know, transcranial focused ultrasound involves the use of ultrasound waves as acoustic mechanical energy that can exert force on neural tissue, both superficially and at deep brain targets, with high spatial resolution, which makes it an important stride beyond other more superficial non-invasive neuromodulation techniques like TMS and TDCS, and allows the possibility of using transcranial focused ultrasound as a non-invasive cognitive prosthetic. In order to attempt to achieve that goal, we sought to build off foundations from the literature. So some of our colleagues at UCLA, Nathia Suthana and Itzhak Fried, namely, have shown in this paper in Neuroscience in 2009 and subsequently in 2012 that deep brain stimulation of the entorhinal cortex and neighboring perforant pathway, which is the afferent white matter projections from the entorhinal cortex to the hippocampus, can improve learning and memory performance in patients with Alzheimer's disease. So this study sought to replicate those findings non-invasively using focused ultrasound. The study was designed as a randomized double-blind trial that was also within subject crossover design. So we had 6 participants, 50% male, 50% female, represented across racial and ethnic lines, who were recruited from the Human Connectome Aging Project that we run here at UCLA. So these were adults who were exemplars of healthy aging, and they were randomized to receive ultrasound of either the left entorhinal cortex, our target region, or the right amygdala, our control region, on their first visit. And they also, and then 2 weeks later, they would receive ultrasound of the target they hadn't received during the first visit, during their second visit. Within each visit, which were basically identical except for which brain region we were targeting, we collected pre- and post-ultrasound neuropsych data, both in terms of learning and memory, both verbal and nonverbal, as well as emotional reactivity data, because we were looking to understand how we could affect amygdala-related behaviors as well. And then in the scanner, which is where we conducted the ultrasound, we collected pre- and post-ultrasound ASL, looking at perfusion, and resting state fMRI. And then during the ultrasound experiment itself, we collected simultaneous BOLD. So this allowed us to have pre and post ultrasound behavior and imaging, as well as simultaneous ultrasound and BOLD imaging for both our target left entorhinal cortex and our control right amygdala, and of course everything from which brain region we targeted to the order of the versions of the tests we gave were randomized and counterbalanced. What we were able to show was that in a single subject, the region that we targeted, here you can see this is the transducer with its built-in fiducials, this was our line of targeting straight to the left entorhinal cortex, resulted in increased perfusion in the targeted region. You'll notice that there was no increased perfusion in the contralateral amygdala, so our increased perfusion region was specific to the area that we targeted. This held up across each individual, and was true at the group level, not only of the entorhinal cortex, but also of the cingulate and medial frontal regions, where we also found increased perfusion associated with ultrasound treatment. We then looked to see if we were having associated functional connectivity changes. So we conducted a psychophysiological interaction analysis looking at what parts of the brain were functionally changing in line with the ultrasound treatment in regions connected to where the entorhinal cortex, where we knew we had increased perfusion. And as you can see across each participant, we had increased functional connectivity in bilateral hippocampi, in cingulate regions, in anterior thalamic regions, and in frontal regions. Many regions known to be associated with the Pepesch circuit or the medial limbic memory circuit, all involved in episodic memory. Unfortunately, we did not find any significant target by time interactions, such that particularly aiming for the entorhinal cortex gave a difference in learning and memory performance. However, we didn't make memory worse. But this was likely related to a small sample size and that we had healthy aging volunteers who were already at the ceiling effect. Moving forward, we demonstrated that these perfusion and functional connectivity changes were also seen in patients with amnestic MCI. And from that work, we've received funding from the Alzheimer's Association to expand this into amnestic MCI and Alzheimer's disease, which is what we're doing now, hoping that with patients with pre-existing memory impairment, we'll have more room to show memory and learning improvement, as well as to demonstrate that these perfusion and functional connectivity changes in a larger group continue to persist in the brains of those with neurodegenerative illnesses, as we've shown already with our preliminary data. We're also considering using personalized 3D holographic lenses that let you create an ultrasonic transducing focus that is a specific shape, for example, the exact shape of a patient's entorhinal cortex, in our studies moving forward. We'd like to thank everyone who's been involved in this: Susan Bookharmer, Martin Monti, the UCLA LifeUp Consortium and Brain Sonics, as well as our Alzheimer's Association Zenith grant. The results of those amygdala, the other side of our study, the amygdala study, are being shown by Bianca Dang and Norman Spivak, and I encourage you to check those out as well. Thank you so much for your time.